Hello, everyone. My name is Yury Gusarov, and today I will guide you through my poster. My poster is called Mitochondrial Mutational Spectrum in Birds, Evidence of Increased Oxidative Damage in Species with High Level of Metabolism. Mitochondrial genome is located within the organelle which produce the majority of cellular reactive oxygen species, or ROS, and thus can be affected by the oxidative damage. The mtDNA mutational signature of Ross is poorly known. Recently, it was hypothesized that AH to GH substitutions on heavy chain, here on my poster, letter H means heavy chain of mtDNA, is an mtDNA signature of the oxidative damage. We observed that this signature is associated with two different life history traits: longevity among mammals, for example, elephants, and environmental temperature among ectotherms. In other words, species with high metabolic rate, like mice. Interestingly, both these factors, longevity and the level of metabolism, negatively correlate with each other. Long-lived species, on average, have lower level of the metabolism, and thus integral oxidative damage should depend on the relative importance of both these factors. But what is the driving factor? That is our hypothesis. We wanted to discover what factor is driving the mtDNA changes, metabolism rate, or longevity. To answer that question, we use our databases of mutation spectrum and nucleotide content of different species based on polymorphic data and database of different birds phenotypes, which we merged and looked for correlations between mtDNA mutational rates and bird ecological characteristics. It's been shown that several life history and environmental traits can be a good proxy of the species-specific level of the metabolism among birds. First, strongly locomotive bird species have higher metabolic level. In this statement, strongly locomotive birds are those birds who are able to fly or run very fast. Second, temperate versus tropical birds have increased concentration of cardiolipins, lipids essential for the activity of the electron transport chains, marking high metabolic rate. Similarly, there are metrics correlating with longevity. Tropical birds are more long-lived and have higher adult survival rates as compared to temperate and Arctic ones, which have a faster pace of life and high reproductive output. Tropical birds have higher concentration of plasmalogens in their mitochondrial membrane, protecting from the oxidative damage. Species with small clutch size typically have increased lifespan as compared to ones who have bigger clutch. In total, we can see that long-living birds, especially tropical, have slower metabolic rate, affected less by oxidative damage, have more chemical compounds which have antioxidative characteristics, like plasmalogens, and have small clutch size compared to their fast-living relatives. To start our research, firstly, using 41 bird species with the reconstructed mtDNA mutational spectrum, we tested the associations between the genomic properties and expected metabolism or longevity of compared species. We observed that A to G on heavy chain tend to be lower in flightless birds, while higher in species with outstanding flight abilities and in the birds with big clutch size. All these results show that highly metabolic birds have increased oxidative damage, marked by the increased fraction of A to G on heavy chain. When we decided to count TSTV, transitions divided by transversions, Results are shown on first bar plot. We use several bird phenotypes. Trop means tropical, wind, wintering, high altitude is high alt, LD long distance migration, NF flightless, DI divers, AI outstanding flight abilities. After estimating TSTV on each species, we observed that birds with outstanding flight abilities have higher TSTV than others. We can see it on figure 1. Also, amount of TSTV is dominating in birds with outstanding flight abilities compared to all phenotypes. We can see it on figure 2. P-value is less than 0.05. Then we observed that species with high GHAH skew tend to have higher clutch size, shorter time of maturation, and higher growth rate. In the end, we made a conclusion that metabolic rate is a greater factor in changing birds' mtDNA structure than longevity. Thank you for listening, and I am waiting for questions on my email. Goodbye.